This is the first of the 3 chapters on pheromones. Pheromones are semiochemicals, which means that they are involved in chemical interaction between the living organisms. We already covered allelochemicals, which are involved in chemical communication between different species. So pheromones are semiochemicals that are involved in chemical interaction between organisms of the same species. Pheromones can be further subdivided according to their function into sex pheromones, defense and alarm pheromones, trail and territorial pheromones, and social regulation pheromones. First 3 classes of pheromones, sex, defense and alarm, and trail and territorial, are all so-called releaser pheromones. That means that they elicit immediate response in recipients. And of course, as you may have guessed, pheromones are defined based on the recipient, not on the producer. The last class of pheromones, social regulation, are so-called primer pheromones. Those often involve a change in hormonal levels and long-term behavior, but they do not elicit immediate response. Based on the chemical composition, pheromones are classified as simple pheromones, pheromones that are composed of only one component, only a single chemical compound. Alternatively, a pheromone may be a complex pheromone that is composed of 2 or more compounds. To isolate pheromones, we apply a number of different methods. Among them, extraction is the most common. Extraction involves treatment of biological material, that means either live or dead organisms, with suitable organic solvent. Then solvent is separated from the material and evaporated, hopefully leaving us a pheromone or mixture that contains pheromones. Extraction may involve degradation of the organism, that means grinding, homogenizing or something like that, or be non-degradative, which means only that organism itself is mixed with the solvent, stirred or rinsed, and then separated. Degradative method is much more efficient because larger amount of material is extracted. But there are drawbacks to it. In the course of degradation, cells are destroyed and compounds of interest may undergo further degradation or reactions. That would give us various artifacts. Artifacts are compounds that are formed later and that are not actually present in the living organism. On the other hand, non-degradative method will result in isolation of less material, but often that material is more pure and presence of artifacts is minimized. Besides extraction, another commonly used method is headspace. There are different headspace techniques, and here on the photograph is a headspace technique as it is applied in perfumery. It's a very good illustration. You can see that what we call a headspace, something like a glass bell, has been placed over the sample, in this case a rose. And with a tube it's connected to the sample collector. Sample may be collected either by cooling to a very low temperature, which means that any gaseous material is going to condense, or it could be absorbed on various absorbent materials such as charcoal or better silica and sometimes alumina. So once headspace is placed over the sample, we drive current of air or inert gas. It could be either driven under pressure or, in the case of the isolation shown in the photograph, at the other end of the tube there is a pump that basically sucks the air in. So as the air is being sucked through the bell, it also takes with it any other volatile materials. Advantage of this method is that it is non-degradative, which means sample is not destroyed in the course of isolation, and of course living organism is not killed, which means that we can do isolation again and again. The drawback is, of course, that usually a very small amount of material is isolated. Once the material is isolated, it has to be analyzed, which means individual components should be separated and characterized so that we can identify them. There are two common methods. One is thin layer chromatography, and another, better instrumental method is gas chromatography. In gas chromatography, or better, gas-liquid chromatography, sample that we have is partitioned between gas and liquid. Liquid is stationary phase, gas is mobile phase. Schematic representation of instrument is given here. So, gas is under pressure, carrier gas is under pressure in a cylinder. It is connected to the injector, which is basically a box, a chamber that is heated to a high temperature. Sample, by means of a syringe, is injected into the injector, and because injector is at a high temperature, it is immediately vaporized. Gas that flows, carrier gas that flows into the injector, will carry the sample into the column. The sample is adsorbed on the liquid phase that is present in the column. Liquid phase is attached or adsorbed to a stationary solid phase, which doesn't do any purpose other than keep liquid phase inside the column. So samples are attached to the liquid phase through intermolecular interaction or interactions, and also they exhibit intermolecular interactions with carrier gas, the mobile phase. Based on the interplay of those intermolecular interactions, they will be flowing through the column at a certain rate. If they attach to the stationary phase very strongly, it will take very long time to flow through the column. On the other hand, if they interact only very weakly with the column and very strongly with the gas phase, they will pass through very quickly. By definition, different compounds must have different set of intermolecular interactions, and if column is long enough, they will be separated by passing through the column. Once they exit the column, they enter the detector, where they are detected and sometimes identified. That depends on type of the detector. We have different types of detectors. The most common and among the least expensive is flame ionization detector, or FID. Basically, sample enters the flame and increase in intensity of the flame is detected. That only detects the amount of the sample and that actually sample is passing through. We don't know anything about its identity. Advantage of this method is that detector is rather inexpensive and extremely sensitive. Similar to it is thermal conductivity detector, which also only detects presence of the sample and its amount, but not what it is. Thermal conductivity detector is a little bit less sensitive than FID, and so it's less commonly used in research. Much better detector is mass spectrometer. As the name indicates, it is a mass spectrometer, so it records a mass spectrum of the compound. Drawback is that it is considerably less sensitive compared to FID or TCD, but the advantage is that since mass spectrum is recorded, if corresponding mass spectrum already exists in the library, compound can be identified by matching the mass spectra. If this is new compound, sometimes mass spectrum itself is sufficient to identify the compound. Finally, another type of detector that is useful in analysis of pheromones is insect antennae. Insect antennae can be connected to electrodes, to microelectrodes, and that would close the electrical current loop. So if insect antennae reacts to a compound, then we'll get a signal. That's very important because insect antennae are very sensitive and often will react only to pheromones. In fact, actually insect antennae will react probably to any compound, but will give very low level signal, and they will give very strong signal when pheromone is detected. We will now examine different classes of insect pheromones. As you have seen earlier on the second slide, insect pheromones can be classified as sex attractant pheromones, defense and alarm pheromones, trail and territorial pheromones, and social regulation pheromones. There are also other pheromones that cannot be classified into any of the above classes, and we simply study them separately as other pheromones. When it comes to sex attractant pheromones, they are the most important and among the best known insect pheromones. To study them, we have to know more about modes of action of sex pheromones. In fact, not just mode of action of sex pheromones, but about different mating practices of different species. First one that we have is mate choice, where members of one sex display to be chosen by the other sex, which means that one sex has a choice. Usually that's female. So usually in species where there is mate choice, that's female choice, where female selects whom she is going to mate with. And that behavior may be governed by pheromones. Mate choice has its own classification. So mate choice may be based on direct benefits. That means that female would be selecting healthy male or male with the most resources. And pheromones that male emits may indicate that male is healthy or that it has a lot of resources. Cantharidin, that we encountered earlier as an antefeedant, may also be considered to be a pheromone, because female may select a male who is capable of producing or providing the most cantharidin. So that would be direct benefits mode of action. Another one is runaway sexual selection, where female or females select the most attractive or the most dominant male. In that case, usually only one or very few males get to mate, and they get to mate with all of the females, while the rest of the males do not get to mate. That's why it's called runaway sexual selection. Only few of the best and the fittest are selected for mating, and they get to mate with all of the females. And finally, third type of mate choice is mate choice or female choice by an indicator. So selection is based on some indicator of good genes, such as size, symmetry, or something like that. So females may select to mate with the largest male, or the most attractive male, most symmetrical, best developed male. So those are three sub-modes of mate choice. Another mode of action of sex pheromones is same-sex contest. In this case, female doesn't have a choice. Instead, members of the same sex, males, fight each other for right to mate. Whoever wins gets to mate with the female. So female has no choice, rather males fight each other, and then whoever wins mates with the female. In this case, pheromones would be provoking aggression in males. Finally, third mode of action of sex pheromones is sexual competition. And sexual competition simply means that whoever gets first to the female gets to mate. So it has nothing to do with fitness or resources or attractiveness or anything else. Whoever is the first, and that may be somebody who was the closest to the female or who was the fastest, so whoever is first to the female gets to mate. That's often the case in species that produce a large number of offsprings. So in that case, producing offspring with inferior genes doesn't really matter. A good example of insect sex pheromone is that of moths. And this is this ester, unsaturated ester shown on the right. It's a pheromone that female emits to attract males. This is obviously a sexual competition pheromone, a pheromone that is designed to attract closest males. So whoever is the closest and is the fastest to come to the female will get to mate. There is really no selection, it's simply mad rush to the female, and whoever gets first will get to mate. It is interesting that same compound is sex pheromone of female Asian elephant, and she secretes it to attract male elephants. Of course, they are not confused, moth and elephants, by each other pheromone, even though it's the same compound. The reason for it is that the amount of pheromone that moth emits is too small to attract elephant. Therefore, elephants won't be confused by moth pheromone. On the other hand, while elephants may emit large amount of pheromone, they are not swarmed by moths. The reason for it is that while elephant pheromone is simple, one component pheromone, just the ester, moth is complex pheromone. There are various species of moths, and to avoid crossbreeding, each species has slightly different complex pheromone. So in addition to this ester, other compounds are present. And since other compounds are missing from simple elephant pheromone, moths are not attracted to it. The second most important class of pheromones are defense and alarm pheromones. We should distinguish between the two. Alarm pheromones cause animals to be excited and look for a possible source of danger. Defense pheromones cause animals to actually take action, usually some defensive action, either to run away or, more likely in case of insects, to attack the perceived threat. Defense and alarm pheromones are common among the ants, and we have here a couple of examples. Example of African weaver ant, which has rather complex 4-component defense and alarm pheromone. That one we are going to study separately in a separate PowerPoint presentation. Another example is that of leaf cutter ant, that has much simpler single component pheromone, that of 4-methyl-3-heptanone. In low concentration, that compound acts as alarm pheromone and causes ants only to be excited and start looking for a source of danger. So they get on their hind legs and start sampling air with their antennae and start moving around. On the other hand, in high concentration, it becomes defense pheromone and they will attack any foreign object. Another species of insects that relies on defense and alarm pheromones are bees. In bees, alarm pheromone is isopentyl acetate. That one only alarms bees, causes them to be excited and look for any foreign objects to sting. However, it doesn't cause them to sting. It is very interesting that this compound is naturally present in bananas and has actually the fragrance of bananas. So bananas will cause bees to get excited. On the other hand, sting pheromone is this rather large, rather non-volatile alcohol. That's cis isomer or Z isomer of 11-icosinol. It makes sense that sting pheromone is so non-volatile. It is deposited together with the sting. And only bees that are in close vicinity to it will be able to notice it, and then they will, since they are exposed to sting pheromone, they will sting the same object or the same enemy. That makes sense because if they were to sting in response to isopentyl acetate, they would be wasting their stings to a number of other objects or organisms that are not a threat, because isopentyl acetate is very volatile and spreads over a large area. Of course, bee can sting only once, so it is very important that its sting is efficient and directed at the right or correct threat. Both of these pheromones, bee alarm pheromone and bee sting pheromone, are themselves complex pheromones, and these are only the major components. There are a number of other minor components contained in them. Another class of pheromones are trail and territorial pheromones. They are used by insects to mark their trail. They are used as recruiting pheromones to attract other insects to the source of food, and they are also used to mark their territory. Ants are well known for their trail and territorial pheromones. Some of them are shown here. You can see that these are compounds of different volatility. Some are very volatile, some less volatile. it is interesting that farnesin, compound shown on the right, is a recruiting pheromone of fire ants. Very similar compound called farnesol is common industrial fragrance, rather nice fragrance, that is added to fabric softeners. Sometimes it may happen that one or more of fire ants mistake farnesol for farnesin and are attracted by it. Then what will happen is, as they are attracted by it, they will lay their own farnesin, actual pheromone, which will attract other ants. The end result is that large number of fire ants could end up being attracted by freshly washed clothing. So, and you can see the result on the left. Another interesting aspect of behavior of the fire ants is that when they attack, they usually don't attack immediately. That's also their attack behavior is governed by pheromones, and what they do is they first swarm. So they first respond to recruiting pheromone and they swarm the intended target, and only then they all start attacking all at once. The pheromones we covered so far were so-called releaser pheromones, pheromones to which response was immediate and obvious. Now we are going to cover social regulation pheromones, at least an example of bees, and those are primer pheromones, pheromones that affect hormonal levels but are not designed to produce immediate response. Queen pheromone of honeybees is produced by queen, or queen's mandibular gland. That pheromone sends message to the hive that queen is alive and all is well. That pheromone lasts for a very short time. First itself, it has 5 components and all 5 are needed to be effective. As they degrade in air, they lose activity very quickly, and also bees themselves actually consume, basically eat this pheromone. So if queen dies, the supply of pheromone stops almost immediately, and then bees start feeding their larvae with royal jelly to rear a new queen. There are other pheromones that don't belong to any of the classes described before. And one example is oleic acid. Oleic acid is that pheromone in bees. It is released upon death of a bee. That's an indication to workers that bee is dead and that should be thrown out of the hive. And bees react to this pheromone so strongly that even if we were to paint a bee with oleic acid and it's a living bee, other workers will try to move it out and kick it out of the hive. There is a rather large database that contains thousands of pheromones, and there is a free access to it, and here is web address. So if you're more interested in more pheromones, this is the web address for it. Now we are going to briefly review physical and chemical properties of pheromones. Pheromones that are used in air in general have between 50 and 20 carbon atoms and molar mass of between 80 and 300. Below the limit of 5 carbons, and even including 5 carbons, and molar mass of 80, there are very few different molecules that are possible. It is necessary for pheromones to be diverse so that each species can have its own pheromone to avoid confusion. So if number of atoms is too small, there are simply too few combinations and too many species would end up having identical pheromones. That's why pheromones must have certain size, above 5 carbon atoms. On the other hand, above the limit of about 20 carbon atoms and molar mass of 300, the molecule becomes increasingly non-volatile. And non-volatile molecules are obviously not suitable as pheromones, as other organisms cannot detect it. Aquatic organisms rely on water-soluble molecules as pheromones. So in that case, it's water solubility that matters and not volatility. Water-based pheromones belong to 3 large classes. They are water-soluble steroids, or large ionic molecules, or finally they could be peptides. Simple pheromone contains only one component, single compound, and that component may be volatile or non-volatile, depending on its desired function. On the other hand, complex pheromone is composed of two or more compounds, and individual components usually have very different volatility. So if you have complex pheromone, you're not going to have complex pheromone of 2 or 3 volatile or 2 or 3 non-volatile chemicals. Instead, there is going to be a range. Some are going to be very volatile and some are going to be very non-volatile. And in complex pheromone, it's usually less volatile or least volatile component that carries the real message, the actual message of that pheromone. Finally, why is study of pheromones important? To us, it's important for a number of scientific, theoretical, and practical reasons. First, study of pheromones helps us understand evolution. Evolution in terms of how organisms adapt to the environment, and also evolution in terms of how pheromones evolved. For example, we have very good indication that sex pheromones have evolved from sex hormones. Another reason why study of pheromones is important is in rearing domestic animals, so we can control breeding of domestic animals. We can also use pheromones in pest control. When it comes to pest control, there are several modes, different modes of use of pheromones. We can do lure and kill and mass trapping. That has been used a lot in the past as has shown to be ineffective. Pheromones could be used to lure insects to a certain area and they're either trapped or killed. Problem is not all of them are lured. And those that survive usually allow insect population to recover very quickly. If they are killed by use of insecticides, large amount of insecticide has to be used because there is large number of insects that have been attracted to the area, and so that pollutes area considerably. They could be used to disrupt mating. So for example, we can use pheromones secreted by female to attract all the males away from the females, and now no males will be available to mate with females, and there will be no next generation. Problem is, again, it usually doesn't work in practice. Some of the males will still be able to reach females, and some will rely on clues other than pheromones, such as visual clues or environmental clues. They will know environmentally where a female should be, where a female is likely to be, and will hang around there and not where we spray the pheromone. The most effective use of pheromones in pest control is in monitoring. That means in monitoring whether certain area has been infested. So we use traps with pheromones simply to check for the presence of the organism. And if that insect is present, if it got trapped, then we now know it's there, and then we can apply pesticide. That way we can avoid unnecessary application of pesticide if pest is not present. That completes this first chapter on pheromones, and now we'll move on to the second chapter. So in the next chapter, we'll look at pheromones of higher organisms.